Morning students. So last class we discussed about that structure of bean seed, that is dicotyledon seed. Alright. So everything what are the parts we have discussed in the last class. So today now we are going to discuss about that diagrammatical representation, that is very important. Draw the diagram and label the part of a dicot seed, that internal structure and external structure. So like that separately it might be asked, or else they may give that diagram and question it may be asked to label the part. So like that everything you have to practice. Alright. So now you see that the structure of a bean seed which is coming under that dicot, because that food material is stored in the cotyledon. Understand? So here that external structure, if we see that the structure of seed, that is the half region of that particular that seed, that is totally that is covered. Then it is, if it is considered to be a seed, here you see that outer covering area, that is what, that testa, we know that the outer seed coat is called as testa. So here if it is seen like that, so that is the outer area, or it is totally covered by one coat, that is what called as testa. Then what about the tegmen? It is not labeled here, just inside, just below that testa, one minute thin layer will be found out over it. So that is what called as tegmen. Alright. Then at the center you see that the funiculus touches with that pod of that particular fruit. That seeds are attached with that pod of that fruit. So that is formed by that funiculus part. So that region after removing that seed from particular that pod, so that one scare-like structure can be found out. So one scare-like structure. So here that region is little bit hot. Some seeds it cannot be seen also, but this bean seed easily it can be seen. So that is what that region is called as hilum. That region, that scare portion where that stalk attaches with that pod of that fruit. So that region is called as hilum. Then nearby that particular hilum, there is a micropores are found out. Understand? So that pores are called as microphyll. Then what are the functions of that microphyll? That is utilized for that exchange of gases and water. Alright. So that is what that external structure. Then when we come to that internal structure, you see when it starts to germinate, when it is getting that favorable conditions, all that conditions like water and air and that sunlight, everything is available, then proper temperature also required. So that is obtained means it will start to germinate. So that is becoming that maturing stage for starting that germination. So at that time, when you see that section of that internal structure of that particular seed, you see that two cotyledons can be found out. So that is what you just take one bean seed and put into the water and just try to separate that cotyledons. It can easily will come out, but separately it may be torn out. Understand? So that can be seen here after taking out that. So two cotyledons where that food materials are stored there. That is why these seeds are called as exoluminous or non-endospermic. Non-endospermic. Understand? So here then that upper region of that embryo axis, and the middle there is that axis will be formed, that is what called as embryo axis, from which total development of that embryo takes place. So there that upper region is called as plumule. Further after germination it is developed into shoot system of that particular plant. Alright, then that lower region is called as that radicle. Then after that radicle is turned into root system. Alright, so further after that germination is started, that radicle is developed as a root system that is growing just below that soil. that plumule is growing just above the soil as a root system. Then we can see that small leaflets will be coming out. So that will be the development of that particular embryo. Then next one, you see, another one region is mentioned here, that is the epicotyle. So there is epicotyle, hypocotyle and that mesocotyle. So in this diagram that mesocotyle is not given. So here what is that mesocotyle? The region where that cotyledons joins with or attached with that embryo axis. That region is a particular region is called as mesocotyle. Understand? That is the center region of that embryo axis. So then now just above that what is formed? Plumule is formed. So in between that plumule and that mesocotyle, that area is called as epicotyle. Understand? So that is what the region or that place between that mesocotyle and plumule is called as epicotyle. Then next one, that hypocotyle. As we discussed earlier, the lower region is called as radicle, that is developed into root system. So now that region between what is there at the center? Mesocotyle. So mesocotyle and that radicle, in between that area is called as hypocotyle. Okay, so that is what that structure of, internal structure of that bean seed. So draw the diagram of external structure and internal structure of that bean seed and label the parts it might be asked, or particular diagram might be given and you may be asked to label that parts. Understand? So next class we shall discuss about that monocot seed, that is the structure of that maize seed. Alright, so go through thoroughly and study properly that labelization, structure of that seed. Next class we shall discuss the maize seed structure. Okay, thank you.